Hi everyone, today I am meeting you here in the Bio 240 Zoom room to go through the lecture about Lab 4, Archaeplastids. And I have some plastids on the screen, my screen behind me here. You might recognize this as a leaf from Elodia. So it's a aquatic green plant, and we can see the chloroplasts and the cell wall in this cell. When we talk about archaeplastids, that chloroplast is an example of a plastid. So it's an organelle that we find in a multi-celled or single-celled or multi-celled organism that has some kind of photosynthetic pigments in it. So those pigment molecules look a color because they are absorbing some wavelengths of light and reflecting other wavelengths of light. We see those colors. So we'll see that the different plants in our plant-like organisms in our group look different colors because they have different pigment molecules. And that we have these eukaryotic cells that can do photosynthesis because they have engulfed into themselves over evolutionary time these what were formerly cyanobacteria that could do photosynthesis by themselves, and they have been incorporated into the cells of these eukaryotic cells. So you'll hear more about that in the lecture. So when we look at the archaeplastids, there's 4 major taxonomic groupings. And we have one grouping, the red algae, and it's also called Rhodophyta. The red algae. The chlorophytes, the green algae. The charophytes, which are also called stoneworts. That won't help you too much because you've probably never heard of them. And then the embryophytes are the multi-celled terrestrial plants that we think of as being plants or land plants. So there are some characteristics as we define this group. They have these plastids that are organelles like chloroplasts. And they're all eukaryotic organisms. And this lineage has as this ancestral algal organism. And we're going to put 3 apomorphies on here that are characteristics that are shared by all the archaeplastids. One of the shared characteristics is that they have chlorophyll A as one of their photosynthetic pigments. So that chlorophyll A is that nice bright green color that we see easily in these green algae, the charophytes, and the embryophytes. So red algae have it too, but those red pigments overshadow it so that you can't really see the green pigments anymore. The other characteristic that they have is they share this cellulose material of the cell wall. So these algae that live in an aquatic environment don't need to stand up against gravity, but they still already had a cellulose cell wall to help support their plant, their body. And then the last characteristic that they all share is called alternation of generations. And that means it's a life cycle where a parent plant that's diploid is going to make offspring that are haploid, and then those haploid offspring are then going to make offspring that are diploid. So they alternate between having an adult multi-celled individual that's diploid versus haploid. We'll come back to that again, and you should also be covering it in lecture. So when we look at examples of the Rhodophyta or the red algae, they are going to have a pretty reddish or purplish, even sometimes dark black almost looking. And so we have some examples here that are just to show you when I say red algae, this is what red algae look like. You're only going to be asked to recognize them by eye, and I will only give you easy, clear ones. The only exception is these coralline red algae, which are shown here. And they are red algae, but they grow a calcified surface on top of their body. So that white calcium carbonate that's layered on top of their red blades makes them look more pinkish, right? So they look hard And pink, you might think they're more like a lichen than they're an algae, but they are actually a live red algae. When they die, they bleach to a white color. And sometimes you'll see those on the rocks if you go down to the seashore as well. The two characteristics that I also want you to know about the Rhodophyta is that they lack flagella. So most kinds of algae have some kind of swimming sperm-like cell to do reproduction with. The red algae don't, so they don't have any kind of cells in any part of their life cycle with flagella. The second important characteristic is that they get this red color from different photosynthetic pigments than chlorophyll A. So there are several different ones. I'm just going to give you one of them, and it's called phycoerythrin. All right, so if you've had your physiology class at all, or you're interested in medicine, you might recognize that erythro part because we use that erythrocytes to refer to red blood cells. So it's basically, it's phycoerythrin, it's a red plant pigment. At the next group we're going to look at briefly are the chlorophyta, the green algae, and they aren't all as lovely and bright green as these examples are. But I'm not going to give you any ones that are tricky, that they look brown but they're green or anything like that. So they take on different body forms, and we might notice that they have a leaf-like structure that's sort of flat and broad. They might form a filament, which is long and thin, and often we're going to see this in single, we'll see a string of single cells, and then they might also have a branched body form where they may have splitting of their stems, and then they end up looking kind of like a tree diagram, right? So codium is an example of that branching growth form. Ulva or sea lettuce on the right-hand side is an example of that flat lettuce leafy kind of structure. So ulva is actually edible. So here are some examples of those green algae, and we'll look at some more examples in lab. The charophytes, there's not very many species of these. They're kind of an odd little group, and they have a new characteristic that's going to define how they look or how they can live compared to the green algae. So when we're thinking about how plants made their way onto land, they needed to have some special adaptations in order to be able to live in the land environment. So it turns out that the first one was with respect to reproduction, that they needed to have some way of having offspring when they're tiny and they're first produced to be able to grow on or to exist in environments that might be dry. So I realized I forgot to put this on this slide. So I'm going to add this characteristic. So between red algae and green algae, there's some difference in characteristics, but we're not going to go into that much detail. But the big one that's important that you know is that this molecule that's used to help protect the spores, and in embryophytes it goes on to become useful in reproduction as well, to protect the pollen grains. We named this molecule quite creatively sporopollenin. And it's a molecule that makes the spores or the pollen able to go into a completely dry environment and not die. So it's a waterproofing tough outer coating. Charophytes have it, red and green algae do not. So these are some plants that grow in freshwater ponds. Typically, they can grow right around the edges. They can get a little bit dry. They can reproduce and have their spores fall onto the edge of a drying pond and not die until the pond fills up again the next time it rains. So these kind of they're tufty looking plants. There's not very many different species. They look like something Dr. Seuss might put into his book. We're going to come back to the embryophytes a little later on and start talking about diversity among the embryophytes. I want to briefly talk about this concept of alternation of generations, though, in a little more detail. So here we have a life cycle. diagram of the alternation of generations. So there's two events in the life cycle that are critical to understanding the difference between different types of life cycles. So we have this alternation of generations for plants. We have a different version of this life cycle for animals and a different version for fungi. We'll go back to those. So fertilization is always going to be defined as the fusion of gametes. So sperm and egg in most organisms. The gametes are single-celled and they're haploid. So they only have one set of chromosomes and their purpose is to fuse with another gamete and form a diploid zygote. So this part of the diagram over here, this is the definition of fertilization. Two haploid gametes come together, their nuclei fuse, they become a single-celled zygote. So the zygote is diploid, it's a different kind of thing than a gamete. So the zygote is going to start dividing and it's going to go through mitosis and it's going to grow into a multi-celled organism. And in the plant life cycle, we're going to call that the sporophyte. So it's a multi-celled, visible to the naked eye, individual. It's diploid. So this is just like an animal body. We're multi-celled, we're diploid. Its reproductive organs are going to be called sporangia. And inside the sporangia are going to be cells that go through meiosis, so like the testes and ovaries in animals. Meiosis is going to be a process that takes a diploid cell and through the special process meiosis of cell division, it's going to make single-celled haploid cells. So the product of meiosis is always going to be single haploid cells. Now in the animal life cycle, product of meiosis is the gametes. Right? So in animals, we go straight across here. Right? We're not going to do that in plants. Animals actually skip a step. In plants, we're going to call those single-celled haploid reproductive structures spores because they're not gametes. They don't do the same thing that gametes do. So spores aren't going to fuse together with anything. They're just going to start to grow by mitosis, and they're going to grow into a multi-celled individual, which can look similar to or very different from the multi-celled diploid individual. So this alternation of generations means we could have the same species of plant, and it could have two completely different kinds of individuals that we might see in the environment that are both multi-celled, but they look completely different. And if we did a karyotype of them, if we looked at their genetic information, we could see that one of them is haploid and the other is diploid. Now that diploid organism, that haploid multi-celled organism is going to be able to produce the gametes, eggs or sperm. It's going to have structures, body organs, that produce those eggs or sperm called gametangia. And in our ferns and our mosses, those gametangia are going to have the names archegonium and antheridium. The archegonium, which has egg in the word, is going to make the eggs, and the antheridium is going to make the sperm. So those are similar to ovaries and testes. So when we look at this life cycle, we have these two different kinds of individuals. They need names to separate them from one another. So the kind of individual that is haploid and it makes the gametes, we're going to call it the gametophyte, and the diploid individual that is going to make the spores, we call it the sporophyte. So its name is based on what it makes. All right. So there's alternation generations that is ancestral to all of our Archaeplastids. But there's a special change that happens in the land plants. And that is that this sporophyte is going to grow as an embryo attached to the mother plant. And so we're going to end up with the sporophyte actually growing out of the body of the gametophyte, just like an embryo grows out of the body of the mother in a human life cycle. However, the human offspring is also diploid, right? So the gametophyte is going to have it's going to be holding the egg on its body, and this is only for the land plants. And it's going to have the sperm come and fertilize it. That zygote is going to start to grow into the sporophyte. And it's going to stay connected to the mother's body and kind of grow out the top of it. So we'll see some examples of what that looks like later. Another characteristic that's unique to the land plants is the body structure called a meristem. We'll start talking about apical meristems. Apical are at the tip. So here we're looking at the tip of the shoot and the tip of the root, very top and very bottom of the plant. Now a meristem is a place in the body, a special location. It doesn't change through life. And this is the place where cells are going to divide. Cells are going to divide through the entire life of the plant. And they're not only going to divide, but they're going to be able to differentiate or turn into all the different kinds of body cells that are needed. So when we talk about stem cells in human biology, we're interested in cells that are going to be able to turn into different kinds of cells in order to help repair some part of the body that's broken. So plants have these meristems, or these locations where they have stem cells for any body part that they might need through their entire life. And this is amazing because you can take a twig off of a tree branch. It's definitely a branch. It's busily making leaves. And if you sort of stick it into the ground and the conditions are right, it'll redifferentiate. It'll start to grow roots right out of that part of the plant that used to be a stem. So that would be like if we started walking around on our hands, that somehow they would know that we needed them to be legs and they would grow legs out of them. It's very weird. So we're going to be able to see this cell division happening. The reason that we look at this region in lab of the root tip here is that that's where the meristem is, and that's where we can see dividing cells. So when we're studying how mitosis works, this is a part of the plant body that we know we're going to be able to find cells doing that. Another characteristic of our land plants is that they have vascular tissue. Now, not all of our land plants are going to have vascular tissue. It's like the idea that we can still have terrestrial organisms that are amphibians. So in a sense, some of our plants, our land plant taxa, the mosses and the the mosses and the liverworts are kind of like amphibians. They need to have water for part of their lifespan. They don't need it for all. So they're kind of in between, right? So this vascular tissue is going to be like having a dry body surface that's protected by scales in animals. And there's also some reproductive changes that happen to allow them to live in a dry environment. So we're going to see when we look at the bodies of the land plants that the ones that are really pretty well adapted to the terrestrial environment are going to have vascular tissue. That means they're going to have the ability to have rapid and targeted transport of body fluids, and that is going to allow them to grow larger. So we already looked at vascular tissue when we did our lab last week as an example of looking at what's inside of a cross-section. I want to draw your attention to another structure on the leaf. So this is a picture of a leaf. It's going to have this vascular tissue going through it. We want to focus on what this, either a cross-section or a longitudinal section through the leaf is going to show us. And we're going to see that we have an epidermis on the surface, that we have some ground tissue cells here in the middle. We have a lower epidermis. We have this vascular tissue over here. And then we have sort of in between these cells in these ground tissue cells, we have air spaces. So in order to do photosynthesis, the cells inside the leaf have to be able to get access to carbon dioxide. And also the leaf needs to get rid of oxygen so that more carbon dioxide can come in. So, and there's also air spaces around the cells inside the leaf so that those molecules of carbon dioxide and oxygen can get to all of the cells that they need to. The way that the oxygen and carbon dioxide go into and out of the leaf is through these structures called stoma. Stoma means mouth, and these are like tiny mouths on the surface of the leaf. So we can see this one down here, and it is an actual opening or hole in the external surface of the leaf that's going to allow carbon dioxide to go in and oxygen to go out. So the stoma itself is the pore, is the opening, and it's created by these two guard cells that are on each side of it. So they're going to look like a little mouth, and there's going to be a little gap in between them that creates that pore or opening, and they can open and close. Right? So when they're in an environment that's too dry, or they're asleep, basically, those two guard cells are going to collapse together. They're going to close that opening. All right, so we're also going to see these in lab today. So when we look at just the embryophytes, so this is the embryophyte tree diagram. And this idea of the transition to land has to do with characteristics that needed to change in order for plants to be able to live on land instead of in the water. So one of them is going to be that is unique to the embryophytes. They have alternation of generation, but they have special body organs for making spores and gametes. Sporangia and gametangia. Now this seems totally normal to us, but if you look at the body of an algae, of the red or the green algae, they don't have a special body organ that's made of some multiple kinds of tissues. They're going to have cells that actually release the gametes or release the spores. They don't have a multi-celled body structure. So having these body organs existing as larger structure and scaling of body, this hierarchy of organization, so that are higher level of organization in their bodies than the algae are. The second important apomorphy for the embryophytes is the embryo. So the embryo grows attached to or on the gametophyte. So the embryo is of the sporophyte. And so there's actually a connection that's essentially the same thing as a placenta in between the growing sporophyte embryo and its parent, the gametophyte. And finally, the other characteristic is this meristem. Right? They have a special place where cell division is going to occur in the body, and it's unique to that location. All right, so we're going to look at 4 major categories of plants and several examples within each one. When we look at the first kind of plants that made this transition to land, they have these basic characteristics and not much else, right? So our examples of moss-like organisms are going to be the bryophytes and the marcantiophytes, and I'll show you what those are. If you're not familiar with these words, botany is a lot of words, and these are ones that might not be familiar to you, but you'll get the hang of it. Then we're going to have these groups, this group of plants that we're going to define as being different from the mosses, the ferns. So when we look at this tree diagram, the difference between the bodies of the mosses and the ferns is that the ferns have vascular tissue. So those very early kinds of land plants weren't really good at being land plants. They didn't have vascular tissue. They're very small. They can't live in very dry environments. And so the mosses, they're like amphibians. Vascular tissue evolved and now exists in the groups of plants that we consider ferns or fern-like. And then also the cone plants and the flowering plants also have vascular tissue. So once vascular tissue evolves, we have much larger plants. We go on to diversify and take over the world, basically. So the fern-like plants, those are going to be plants that are in the groups Pteridiophyta and Lycopodiophyta. Never heard those words? We'll look at some examples in a few minutes. So the moss is going to be a group that has no vascular tissue, And the ferns, cone plants, and flowering plants are going to be in a group that has vascular tissue. And the other thing that is different about cone plants and angiosperms is that they both have pollen and seeds. So mosses and ferns neither have pollen nor seeds. And so that's an important characteristic. The evolutionary invention of pollen and seeds made the land plants completely terrestrial. So if we think of a comparison between amphibians and reptiles, the thing that makes reptiles truly, truly terrestrial compared to amphibians is that the reptiles can lay an egg on land, on dry land. It has a hard shell that's waterproof and protects it from that terrestrial environment. Same thing is true of pollen and the seeds. They have those sporopollen molecules around the pollen. The seeds have a seed coating, and those are both reproductive structures. So the equivalent of pollen and seeds for land plant evolution is like the amniotic egg is for tetrapod evolution, right? So mosses and ferns are like amphibians, and then everything on up is more like reptiles on up. All right, so cone plants, gymnosperms, we're going to look at three kinds: the pinophytes or conifers, cycadophytes and ginkgophytes, and then the Magnoliophyta, is all the flowering plants. So they're going to be characterized by having flowers and fruits. Flowers and fruits. There should be 2 stars on the diagram. And then the 2 major groupings within the Magnoliophytes are the monocots and the dicots, and we looked at some of those characteristics last time. All right, so let's take a tour through these different kinds of plants. The bryophytes are true mosses. In our lab manual, we're going to look at some slides, and those are going to be moss is neem, and it's a little tiny, tiny green, fluffy looking plant. So they're really tiny. They don't have a lot of variety in how their bodies look, and today we're just going to look at a moss body and be wowed at how tiny and cute they are. We're only going to find them growing in moist habitats, so they're not very diverse or common around San Diego, although we do find them in areas that are moist. They'll grow during the winter, and then they'll die. They'll leave their spores around so that they can grow again the next season. Archaeplastids are liverworts. Liverworts get their name because when we look at the shape of the body of these plants, they tend to have these wide, flat, overlapping lobes. And if you've ever done a round dissection, you know that the liver has these wide, flat, overlapping lobes. So that's how they get their common name. These are also fairly small plants. They tend to grow in sort of mats. They're not more than a few centimeters high, and they have this flattened looking leaf. And again, we have one example that we're going to see in lab. So those are our non-vascular seedless plants. Now we're going to start looking at the vascular seedless plants. So the vascular seedless plants includes 2 groups. The Lycopodiophyta, and we have 2 examples of different species that we find in the Lycopodiophytes. We have these ones called spike mosses. Now I put moss in quotes here because they're not true mosses, they're not bryophytes, but they're sort of fuzzy looking, so people call them mosses. That's the problem with common names. They can be descriptive but not taxonomically relevant. All right? So we have this plant here, Selaginella, is our example of a spike moss. And so what you need to know is you need to recognize it. You need to know that it is an example of a Lycopodiophyte. You need to know that it is a seedless and a seedless vascular plant, a vascular plant. So notice the size and shape of the leaves that it has here. They're still small. Like the moss leaves are really small, but these Lycopodiophyte leaves are also small. Other grouping within the lycopodiophytes are also confusingly called club mosses. And our example is Lycopodium. The Lycopodium is kind of looks like tiny little Christmas trees, maybe. And if it has a reproductive structure, it's growing like out of the tops, we can see in this picture. So these are a little, quite a bit larger than mosses and liverworts. And, but they still don't get as big as the Unisea Lycopodium trees, right? And they do not have flowers or fruits. Again, I just want you to recognize this specimen. Know it's a lycopodiophyte. Know that it is a seedless vascular plant. So what I mean by these plants being vascular or non-vascular, we're going to be able to notice that when we're looking at the cross-section of their stem. So here are cross-sections of the stems of a moss, right, which is non-vascular. And then Selaginella, Lycopodium, and Psilotum. We'll see Psilotum in just a minute. It's a fern-like plant. So remember, we have two major clades within the vascular seedless plants. We have the Lycopodiophytes and we have the Pteridiophytes, the fern-like plants. So Psilotum is an example of a Pteridiophyte. We'll see pictures of it in a minute. So I want you to look at the cells of these cross-sections of the plants and look at the arrangement of them. You can see vascular tissue easily in these plants that are here. So these are vascular tissue, this is vascular tissue, this is vascular tissue, and we see these large open cells with a thick cell wall. So we can identify those as a vascular bundle, right? When we look at this moss stem over here, and we look really carefully at it, we don't see those dark staining large open cells. Especially we're going to look near the middle of the stem because that's where those vascular bundles are in these relatively closely related plants. We do see a little bit of darker cells towards the outside of the ring, but they're not bigger than the rest of the cells around them. And so we're going to know that the plant cells are not, that these sort of darker stained cells, they're not vascular tissue because the middles of them aren't bigger than the surrounding cells. What they are is for structural support. So they have a little bit of reinforced cell wall that's a little tougher and stronger. And they're going to be able to live on land because of that. All right, so we have three different kinds of cells that we find in the tissues of plants, and I'm briefly going to tell you what they are right now because it helps us identify when we're looking at different types of tissues. So these really kind of open, plain, boring looking cells that are part of the ground tissue, these are called parenchyma. And if I was going to draw what one of these cells look like, it would be moderate size, thin cell wall. Turns out these cells that are more around the outside, they are for structural support and they are called collenchyma. And if we drew a close-up of those cells, they would have typically a somewhat smaller inside and then a thick cell wall. Now, when we look at our vascular tissue cells, compared to the cells around them, they're going to be larger and they're going to have a thicker cell wall, and it's actually going to be made of a different material. So it doesn't necessarily have a super thick cell wall, but these sclerenchyma cells have a material in the cell wall called lignin. Right, now remember that our cell walls in plant cells, we learned the basic idea of what they have is cellulose. So we have a cellulose cell wall in the parenchyma cells. The sclerenchyma cells have lignin as well as cellulose. So this woody tissue is this kind of cells are going to be what the woody tissue in those plants, trees, and shrubs that have a brown type of cells. These cells are dead and empty at maturity. So these cells are actually dead. The collenchyma and parenchyma cells stay alive. Right, so there are these vascular tissues. The only reason we can pass the plant body fluid through them is because they're empty, they're hollow, they're just like pipes or tubes. Some kinds of sclerenchyma cells are alive, but the ones that are part of the vascular tissue are not the xylem cells. All right, so we see that these cells that stain, all right, so the reason they stain different colors, these ones that have these thicker cell walls are going to stain with the different from the parenchyma cells. So we can see that these cells that are surrounding the moss stem, even though they're staining darker, it doesn't mean that they're vascular tissue, right? So we can look at them carefully and we can see that they are for structural support, they're collenchyma cells, and they are not vascular tissue. So take home message, moss, non-vascular plant. We look at a cross section of its stem. We indeed do not see that it has any vascular bundles anywhere. We can convince ourselves indeed it does not have vascular tissue. So all these other plants do. So the lycopodiophytes, the pteridiophytes, the fern-like plants, we can clearly see this vascular bundle, and that it's all the way in the middle of the cross section. Another characteristic that differs between lycopodiophytes and other plants is that these more primitive plants only have what are called microphyls. That is, they have little leaves. Each leaf only has one little vein extension in it, and they're overall very small. When we look at plants that are larger, what ends up happening over evolutionary time is these multiple tiny little microphyls have fused together, and so we have leaves that can be much larger. They have multiple extensions and branching patterns of the veins inside them. So starting with the ferns, the pteridophytes, we're going to see megaphylls, and that allows a much higher capacity for photosynthesis. All right, our pteridiophytes, there's quite a bit of diversity amongst them. And they're, a lot of these plants are going to be these ferny looking plants. So the most diversity, they live in moist habitats. They can stand a lot more drier habitat though than the non-vascular plants. And the Polypodiopsidae are the true ferns. So when we think about a ferny looking plant, it's probably one of these. So Polypodium is a really common house plant, kind of fern plant. And it's we're going to call these the true ferns. And when they grow with these little fern curlicues that are called fiddleheads. So that's going to be one way we know we are looking at a true fern. If we can see some baby leaves growing and we see them doing that curlicue. Another group within the pteridophytes are the whisk ferns. So if you think about a whisk broom, that's one that you're going to be able to brush some crumbs out of the kitchen floor with. It's kind of a small broom. So if you imagine taking one of these Psilotum plants and turning it upside down, you could actually, whisk with it. This is it. This is what Psilotum looks like. All 15 species look basically like this. Their body branches in two. They look like a cladogram. They don't have any large sticking out leaves. And when they do become in a reproductive condition, they get these little swollen parts on their stem. And we'll see some slides of those next week. So Psilotum, again, it's an example of a vascular non-seed plant. I would like you to know that it's in the same phylum as the pteridophytes and that it's in the Psilotopsidae. Our third group within the pteridophytes is the Equisetapodes, Equiseta. Such a problem. Excuse me. All right. And these are the horsetails. Yes, I know. Sorry. Horsetails kind of look like horsetails. There's not a lot of diversity in their visual structure. Again, they kind of look like maybe asparaguses. They're sticking up, and then they have maybe some kind of frilly stuff. If they do have a reproductive structure, it's a cone-like thing that's sticking up. They do photosynthesis on their stem, so the stem looks green, and they can be quite large. They can be, head high, but they're these weird single-looking stalks. So I just want you to know what group these are in. Equisetum, right? Horsetails. Equus is the genus of horses, if you are a horse person. All right, when we start looking at the plants with cones, we're going to look at the plants first that only have cones, right? The cone-bearing plants. And our first example is the ginkgo phyte. There's only one species, ginkgo. It's a tree. It's a beautiful landscaping tree. It has these characteristic band-shaped leaves, quite beautiful. And it's only one species. It looks like it has some kind of berry on here. That's actually a cone. And these guys sort of look like they might be flowers, but they're a cone. And so if you recognize this tree and you say that it's a ginkgo phyte, it is a vascular plant with seeds and pollen. But what does it not have? It doesn't have flowers or fruits. Our next group is the cycads. Now we have one that's commonly used in landscaping around San Diego, and it's called a sago palm, right? Again, palm is in quotes because it's not a palm tree. Palm trees are actually flowering plants. They're monocots. And what's going to be a characteristic of our cycad is that it can have kind of a stalk, and then it's going to have a middle, and then it's going to have these stiff leaves radiating out around it. They're going to be a little ferny looking, right? It can also grow these sort of stalks up out of the ground and have even more ferny looking leaves on them. There's always going to be an empty space in the middle. And the empty space in the middle, sorry, this looks like a sea anemone more or less, is because that's where these reproductive structures are going to grow. So this is a cone. There's a female cone. These are cone plants, remember. Seeds are going to grow out of it. This is a female cone. And this one over here that's long and thin and tall is a male cone. And it's going to make the pollen, right? So there's a tremendous diversity of cycads at the San Diego Zoo. And I have lots more pictures of them for you in the supplemental tutorial for this slideshow. All right, I know we've got a lot of groups here. We're going to go back over them again in the next lab. So I want you to just focus today on recognizing what they look like and being able to tell them apart from one another. We'll come back to more of their reproductive. Right, our last two major taxa are the Pinophyta, or what used to be called the conifers, pine trees, basically. So these trees are going to be woody trees. They can be very tall, our tallest kinds of trees, and they're going to be characterized often by having a body form that grows like if a little kid were drawing it in a triangle. We can also see through them. Their leaves are not completely filling in all of this space. So if you look here, you can see through this tree. You can see through this tree. Those are those nice places to hang those Christmas ornaments. And we use these trees in the winter because they don't drop their leaves off of them. So they're going to have a characteristic of being very hardy, able to live in more challenging circumstances than the flowering plants do. And so we'll often find these at high elevations or at latitudes where the winters are very long and harsh. They come in two slightly different body types. We're going to see that they have needle-shaped leaves. That means that the leaves are long and thin. Sometimes they're even a little pokey at the tip. And these kinds of trees include things like pine, fir, spruce, cedar, cypress, redwood, sequoia. The other kind of body form in the pinophytes have scale-shaped leaves. So they have overlapping like the scales on a fish. They do form these sort of twig-like shapes, but if you look very closely at them, they're not needles. They have more of a scaly-looking surface. So these kinds of plants are also very common in San Diego area landscaping, and they are things like junipers or cypress plants. And many of them are shrubs rather than trees. All right, the Magnoliophyta, the flowering plants. The vast majority of plants that are in the Archaeplastids, over 250,000 plants, are the flowering plants. And within the flowering plants, the vast majority are the dicots. So we did a quick introduction to monocots versus dicots previously. So when we talk about monocots, those are going to be plants that are things like we call lilies, tulips, palm, grasses, orchids, bamboos, and we have some examples on that left-hand side. And then the dicots are going to be all other kinds of plants. And we have examples of poppy, of basil, of hibiscus. And so we'll come back to another lab and do the whole lab. So we'll look in much more detail at them because they are so diverse. All right, that was our quick introduction to the Archaeplastids. So remember, that includes the red algae, the green algae, the charophytes, and the land plants. And then the land plants, we've got those non-vascular seedless plants, the mosses, Bryophyta, and the liverworts, the Marcantiophyta. We have the vascular plants with no seeds. Those include the Lycopodiophytes and the Pteridophytes. Then we have the plants with seeds and pollen, the cone-bearing plants, Ginkgophytes, Cycadophytes, and Pinophytes. And then we have the flower and fruit plants, Magnoliophytes. All right, we will come back and go over more of the information about these groups in the next two labs. I want you to start working on recognizing them and knowing what their major evolutionary characteristics are for this lab.